Welcome to Gnana Deeksha, a concept-based digital learning. I am Dr. Swaroopa Rani, lecturer in microbiology from Telangana Social Welfare Residential Degree College for Women, Mahendra Hills. Today, we are going to learn an important and befitting topic for the current pandemic, that is vaccines. It's high time that every one of us should know about vaccines, as we are aware that prevention is better than cure. It can happen only when we have a vaccine for specific disease. After this session, we will learn about the origin and development of vaccine, different types of vaccines and their route of administration. Also, it explains how vaccines work and the importance of vaccination in providing herd immunity. Finally, we will have a glance at the stage of vaccine development for current COVID-19 pandemic is. Let's understand what is the need for vaccines. When treatment is available for many of the diseases, the first major breakthrough in the treatment of infectious diseases came from the introduction of antibiotics. But as you can see in this picture, soon after the use of antibiotics like penicillin, streptomycin and chloramphenicol etc. developed resistance in very short span. Also, the antibiotics can destroy only certain types of bacteria and they are not effective on the infectious agents like viruses. This makes preventive vaccination as the only alternative for many of the infectious diseases. Moreover, most of the existing vaccines, except very few, have not shown any resistance even after prolonged usage. Now, let's first look at the history behind the development of vaccines. The first recorded attempt was made by Chinese on Turks in the 15th century to induce immunity to smallpox, a dreadful disease caused by the virus variola major, which has 30% mortality rate and survivors are often left with disfiguring scars. They have introduced dried crusts from smallpox lesions either by inhalation or through small cuts in the skin, a technique called variolation. In the year 1718, the positive effects of variolation was observed by Lady Mary Montague, a wife of British ambassador, and she performed this technique on her own children. Later, in the year 1798, a historical development in the field of immunology was made by an English physician, Edward Jenner. He observed that the milkmaids who contracted with cowpox virus that causes a mild disease were immune to smallpox. As you can observe in this picture, he tested his idea by introducing fluid from cowpox pustule into an 8-year-old boy. And later, he intentionally infected that child with smallpox virus. And as predicted, the boy did not develop the smallpox. This was the major breakthrough which led to the development of vaccines. And Edward Jenner is being considered as father of immunology. The next major advance in the field of immunology is the development of immunity to cholera in chickens. In 1878, Louis Pasteur succeeded growing the bacteria that causes fowl cholera such as Pasteurella multocida. After returning from his vacation, he and his team continued their experiments and injected some chickens with same old culture. And the chickens got ill, but surprisingly they recovered very soon. Later, Pasteur grew a fresh culture for injecting it into new chickens, but due to the limited supply of chickens, he had to use same old chickens along with the new ones. But to his surprise, only the fresh chickens died and the old chickens survived and were completely immune to the disease. This led to the discovery of attenuation, that is, the aging of the culture had weakened the virulence of the pathogen and upon administration it induced immunity. And he called this attenuated strain as vaccine in honor of Jenner's work with cowpox. Later, he applied this technique for developing other vaccines such as anthrax for sheep caused by Bacillus anthracis and rabies for humans caused by rabies virus. Pasteur administered his first human vaccine to a young boy named Joseph Meester, who had been repeatedly bitten by a rabid dog, and the boy survived. Now, let's understand what is vaccine. Vaccine is a biological preparation that provides active acquired immunity against specific diseases. A vaccine typically contains an agent that resembles disease-causing microorganism and is often made from the killed or attenuated microorganisms or their toxins or their surface proteins. Either they are introduced by mouth, by injection or by nasal spray to stimulate the immune system and recognize the foreign agents and destroy them without causing any severe disease. Further, they even have the memory to recognize in future encounters by same pathogen and destroy them. Before we enter into the details, let's first look at what are different kinds of immunity. Immunity is classified into two types. One is innate immunity, which comes from birth, and the other one is acquired or adaptive immunity, is developed during the person's lifetime. Acquired immunity is again of two types, active and passive. Active immunity is either natural or artificial. If a person acquires an infection by natural exposure to any pathogen via food, water, air or any other source, our body develops immunity to protect from such diseases. This kind of immunity is called natural active acquired immunity. In contrast, if a person develops immunity to artificially acquired antigens in the form of vaccines, it is called artificially acquired immunity. As we have seen in the definition, vaccines work by providing active acquired immunity, whereas passive immunity is the transfer of ready-made antibodies rather than involving our own immune system, and it can also occur naturally or induced artificially. If the transfer of antibodies from mother to the baby occur through the placenta or by breastfeeding, it is called natural passive acquired immunity. And if the transfer of preformed antibodies to the non-immune person takes place, then it is known as artificial passive acquired immunity. We will discuss this in more detail in the following sections. Now, let's learn different types of vaccines that currently exist and in developmental stage. There are several vaccine classes, each with unique advantages and challenges, which includes live attenuated vaccines, inactivated or killed vaccines, polysaccharide vaccines, toxoid vaccines, recombinant antigen vaccines, recombinant vector vaccines, DNA vaccines and RNA vaccines. Let's see one by one. Live attenuated vaccines contain attenuated or disabled microorganisms which lost their ability to cause significant disease but retain their capacity to induce the immune response. Attenuation can be achieved by growing a pathogenic bacterium or virus for prolonged periods under abnormal culture conditions. For example, vaccine for tuberculosis such as Bacillus calmette-guerin was developed by growing Mycobacterium bovis by 239 serial subcultures in glycerin bile potato medium over a period of 13 years. Also, Sabin oral polio vaccine that included 3 attenuated strains of polio virus. Other examples for live attenuated vaccines include MMR, chickenpox and rotavirus. Whereas killed vaccines are made from microorganisms that have been killed or inactivated with chemical or heat treatment. but still they can induce immune response. In particular, the Salk polio vaccine, that is injectable polio vaccine, is produced by formaldehyde inactivation of the polio virus. Examples include influenza, cholera, plague, hepatitis A and rabies vaccine. Both the vaccines have some advantages and disadvantages. Live attenuated vaccines generally require only one dose to induce long-lasting immunity, whereas killed vaccines often require repeated booster doses. Live attenuated vaccines elicit strong immune response by inducing both humoral and cell-mediated immunity, while the killed vaccines are less effective as they can only induce humoral immunity. However, killed vaccines are more stable and safe than live vaccines, as these live forms may mutate and revert to virulent forms that results in disease. To avoid the risks associated with these vaccines, a strategy was developed where only specific purified molecules such as polysaccharides or toxins or recombinant proteins derived from the pathogen are used. These are called subunit vaccines. Now, let's look into subunit vaccines. Some pathogenic bacteria have polysaccharide capsule around their cell wall, which is primarily responsible for the virulence. The vaccine for Streptococcus pneumoniae PPSV23 consists of 23 antigenically distinct capsular polysaccharides, and Neisseria meningitidis vaccine also consists of purified capsular polysaccharides. Polysaccharide vaccines can induce only limited immune response by activating B cells in T-independent manner. As they cannot activate T helper cells, results in only IgM production but with little class switching and no memory. However, vaccines that conjugate a polysaccharide antigen to a protein carrier can induce the stronger immune response by Th cell activation. This image shows the linking of toxoid protein carrier to polysaccharide component of capsule. Let us take few examples. Haemophilus influenza type B Hib vaccine, it is covalently linked to tetanus toxoid, and type B TCV typhoid conjugate vaccine is also linked to tetanus toxoid, while meningococcal vaccine MCV4 is joined to the highly immunogenic diphtheria toxoid. Second class of subunit vaccines include toxoid vaccines. Some bacterial pathogens including tetanus and diphtheria causing bacteria such as Clostridium tetani and Corynebacterium diphtheriae produce exotoxins that account for most of the disease symptoms. Vaccines for these diseases are made by inactivating the purified exotoxin with formaldehyde. Vaccination with the toxoid induces anti-toxoid antibodies which are capable of binding to the toxin and neutralizes them. Examples: diphtheria and tetanus toxoids, which are given in combination with pertussis vaccine as DTaP or DPT. Another class of subunit vaccines are recombinant antigen vaccines. The gene encoding any immunogenic protein can be cloned and expressed in culture cells using recombinant DNA technology. A number of genes encoding surface antigens from viral, bacterial and protozoal pathogens have also been successfully cloned for vaccine development. Hepatitis B vaccine is first such recombinant antigen vaccine approved for human use. This is produced by cloning the gene for the major hepatitis B surface antigen and expressing it in yeast cells. The recombinant yeast cells are grown in large fermenters to accumulate HBs antigen in the cells then the HB vaccine is extracted and purified for commercial use. Other vaccines currently under development include recombinant vector vaccines, DNA vaccines and RNA vaccines. As we have seen that though the live attenuated vaccines induce strong immune response, they can sometimes revert to pathogenic forms. This disadvantage can be overcome by the use of recombinant vector vaccines. Individual genes that encode key antigens can be introduced into vectors such as attenuated viruses or bacteria and they express the gene product in the host. Since most of the genome of the pathogen is missing in these vector vaccines, it cannot revert back to original pathogenic forms. A number of organisms have been used as vector vaccine preparations such as vaccinia virus, adenoviruses, etc. DNA vaccines or third generation vaccines. They contain plasmid DNA that encodes specific antigenic proteins from a pathogen. The DNA is injected into the body and taken up by cells to synthesize proteins in vivo. DNA vaccines have potential advantages over conventional vaccines. They can induce both humoral and cell-mediated immunity. Currently, several DNA vaccines are available for veterinary use and human trials are underway for malaria, HIV, influenza, Ebola, herpes virus and for several cancers. RNA vaccine is a new type of vaccine which works by introducing an mRNA sequence into the body. This sequence is translated to produce the encoded antigens which then stimulate the immune response. RNA vaccines are faster and cheaper to produce than traditional vaccines. Besides, they are safer to use. Most contemporary research is into RNA vaccines for infectious diseases, cancer, allergies and current COVID-19. Now, we shall have a look at different components of vaccines including antigens, adjuvants, antibiotics, preservatives and stabilizers. Antigens are the active components derived from the structure of disease-causing organisms which induce immune response. Adjuvants enhance immune response to vaccines. Antibiotics are used during the manufacturing phase to prevent bacterial contamination. Preservatives are added to multi-dose vaccines to prevent bacterial and fungal growth. Stabilizers are used to help the vaccine maintain its effectiveness during storage. In addition, vaccines also have some residual byproducts from the production process. Let's understand the role of adjuvants in the vaccines. These are the substances that are added to vaccine preparations to enhance the immune response to the antigen or pathogen with which they are mixed. This is especially important in case of killed or subunit vaccines where immunogenicity can be quite low. The commonly used adjuvant in human vaccines is aluminum salts called alum and is primarily thought to work by creating slow release and delivery of the immunogen at the injection site which helps in sustained stimulation of the immune response. Other adjuvants used are AS04, MF59, AS01B and CpG1018. The route of administration is the path by which a vaccine is brought into contact with the body. This is a critical factor for success of the immunization. A substance must be transported from the site of entry to the part of the body where its action is desired to take place. This picture describes the different routes employed for the various vaccines administration. Intramuscular injection administers vaccines into the muscle mass. Vaccines containing adjuvant should be injected intramuscularly to reduce adverse local effects. Subcutaneous injection administers the vaccine into the subcutaneous layer, and intradermal injection administers the vaccine in the topmost layer of the skin, whereas oral administration administered orally. A recent route of administration is intranasal spray, a new vaccine approach. Let's understand the mechanism of vaccines. Vaccines work by inducing both branches of adaptive immunity, humoral and cell-mediated immunity. When our body exposes to the antigen for the first time, it is engulfed by the antigen-presenting cells and presented to T helper cells. T helper cells will get activated and produce cytokines, which in turn activates B cell. Activated B cell differentiates into plasma cell and memory cell. Plasma cell secrete antibodies which binds to the specific antigens on the extracellular pathogens and neutralize them, or make them better targets for phagocytic cells and complement proteins. In cell-mediated immune response, the antigen is presented to cytotoxic T cells and it gets activated by T helper cell. TC cell differentiates into effector cytotoxic T cells and memory T cells. Effector CTLs bind and initiate the apoptosis of infected cells and also cancer cells. When body exposes to the same antigen for the second time, memory B cells produce large number of antibodies with much more efficiency. The unique attribute of adaptive immune system is immunologic memory. The immune response generated during first encounter to antigen is termed as primary immune response, and subsequent exposure is called secondary immune response. In primary response, IgM predominates and peaks at 7 to 10 days, but in the secondary response, IgG predominates with a peak of 3 to 5 days. The secondary response is both faster and stronger than the first. Vaccines imitate natural infection and induce the immunity. Now, let's see the journey of vaccines. Before a new vaccine is given to people, it must go through several stages of testing to make sure that it is safe and efficient. In discovery and preclinical phase, researchers work out to develop a potential vaccine for the organism and test it in the laboratory and on model animals. In clinical phase, Phase 1, the vaccine is first tested on a small number of healthy people, around 20 to 100, to check that the vaccine is safe and immunogenic. In Phase 2, the vaccine is then tested on several hundred people and the immune response is studied in more detail about its immunogenicity and safety. In Phase 3, the vaccine is tested in thousands of people to see the efficacy, immunogenicity and safety. The Central Drugs Standard Control Organization (CDSCO) licenses a vaccine only if it is safe and effective. Also, benefits outweighs risks. Now, let's move on to the benefits of vaccines. Vaccines are safe to use, support the immune system, provide protection against infectious diseases, protect future generations, prevent unnecessary medical costs and save from complications of the disease. Usually, vaccines are considered safe and effective. However, vaccines are not risk-free. Occasionally, adverse event will occur following vaccination. Common and minor reactions include pain, swelling, redness, fever and malaise. Rare and usually more severe reactions include anaphylaxis. Nowadays, we are frequently listening the term herd immunity. A mass of people acquire protective immunity either through vaccination or natural infection. This principle is called herd immunity, which works by decreasing the number of individuals who can harbor and spread an infectious agent significantly decreases the chance of susceptible individuals to get infected. This picture clearly demonstrates how herd immunity works. Here black color indicates for infected people, blue for unvaccinated, and yellow is for vaccinated individuals. As the percentage of vaccinated is increasing, the chance of spread of disease is decreasing. When only some people get vaccinated, disease can spread rapidly through a population. But when enough people are vaccinated, contagious diseases can be kept from spreading and the opportunities for an outbreak of disease become so low. Even people who aren't immunized benefit. Herd immunity is very critical because there are always some individuals who cannot receive the vaccine. For example, very young children or immune compromised individuals. Vaccination is the process of vaccine administration to immunize people. This table represents the National Immunization Schedule. The best gift we can give to our child is proper vaccination. It is vaccination, not vaccines, that saves life. So, don't forget to immune your child. Vaccination is the process of vaccine administration to immunize people. This table represents the National Immunization Schedule. The best gift we can give to our child is proper vaccination. It is vaccination, not vaccines, that saves life. So, don't forget to immunize your child. Vaccines are one of the greatest inventions in the human history. During the last 2 centuries, vaccines have eradicated smallpox, reduced global child mortality rates, and prevented countless birth defects and lifelong disabilities. Through successful vaccination programs around the world, polio is almost gone. We are very close to complete eradication since 2 wild polio strains has been eradicated recently and only type 1 strain exists now. Also, you can see in this picture, vaccines has drastically decreased morbidity of many of the diseases in the population. As we have discussed in the previous sections, immunization can be achieved by both active and passive immunization. Emil von Behring and Kitasato were the first to show that immunity elicited in one animal can be transferred to another by injecting serum from the first, for which von Behring was awarded with Nobel Prize. Before vaccines and antibiotics became available, passive immunization was the only effective therapy for some fatal diseases such as diphtheria and tetanus. Passive immunization can be achieved naturally by the transfer of antibodies from mother to the fetus and artificially by injecting pre-formed antibodies from immune individuals to recipients. It provides only temporary immunity without the development of memory B or T cells. Presently, in plasma therapy, antibodies from completely recovered patients known as convalescent donors is being explored as potential therapeutic intervention for current COVID-19. COVID-19 has been one of the toughest challenges we have ever faced. Across the globe, everyone is waiting for the vaccine. Let's see the world's progress in the hunt for COVID-19 vaccine. Researchers worldwide are working around the clock to develop a vaccine. There are currently more than 170 COVID-19 vaccine candidates under development. 35 of them are in clinical trials, 9 of them entered phase 3. In India, Covaxin of Bharat Biotech and Zycovir of Zydus Cadila are at completion of phase 2 trials. Let's summarize what we have learned today. Vaccines have the greatest potential to promote public health. They reduce morbidity and mortality from infectious disease, save costs as well as lives. Vaccines mimic the pathogen and induce immune response in the body. There are 6 types of vaccines currently used or under experimental consideration in humans: live attenuated, killed, subunit, recombinant vector, DNA and RNA vaccines. Adjuvants are the substances added to vaccine preparations that helps in delivery of the vaccine and enhance immune response. There are 4 major routes of administration of vaccines, and they work by inducing both humoral and cell-mediated immunity. We have seen the journey of vaccines, how they are regulated from development to licensure to use by national regulatory authorities. The risks associated with vaccines are very low compared to the risks of the diseases they are designed to prevent. Also, we have seen the importance of vaccination in providing herd immunity that works by protecting entire population if enough people get immunized. In addition to the active immunization, we have learned about plasma therapy, which is a passive immunization, the only effective therapy in some fatal diseases, including some cases of COVID-19. Finally, we have looked at the race for COVID-19 vaccine. Some of the potential vaccine candidates entered phase 3 trials and some are in phase 2. Vaccination is one of the greatest public health achievements of medical history that has saved many lives. Besides current COVID-19, it is evident from this picture for many more infectious diseases there is no vaccine available yet. It's clear that new vaccines can be a breakthrough for global public health. In order to keep pace with new threats, research and development must continue with the same intensity that we have seen in recent months. Until we find a vaccine for COVID-19, everyone should follow the guidelines for preventing the spread of coronavirus. No one is safe until everyone is safe. Thank you, one and all.